Hi everyone, today we're going to be talking about DNA structure as well as some important terms to know for DNA. So we're going to start by talking about the bases of DNA that exist. So I'm sure you have heard about these already, but we have cytosine, thymine, adenine, and guanine. And cytosine and thymine are considered pyrimidines, meaning that they have only one ring, whereas adenine and guanine have two rings, and these are considered purines. And the way I kind of remember this is something that works for me is I notice that C and Y both have, cytosine and thymine both have Y's in them, and so does pyrimidine. So that's how I remember that these are all together as one group. And then purines are spelled kind of similarly to adenines and guanines. And so I just know that they're also one group according to the fact that they don't have the Y's in them. So that's how I remember it. I'm sure you have your own ways of remembering as well. But one has one ring and one has two rings. Nucleotides are considered the building blocks of DNA, and they're composed of a phosphate group, a pentose sugar, in this case it's deoxyribose for DNA, and the reason it's deoxyribose is because you would imagine that there should be an OH over here, hydroxyl, as there is in ribose, but in deoxyribose that OH is no longer there. And then the last component is a heterocyclic nitrogenous base. And just to clarify, the heterocyclic term here basically refers to the fact that the atoms that compose the ring are different types. So this ring is composed of different types of atoms. So for example, this one has carbon and nitrogen making up the ring. And the last thing I want to highlight on this slide is something called a nucleoside. And nucleosides are basically this part right over here. So it basically is composed of your pentose sugar and your base, but excludes the phosphate group. So the nucleoside is just the sugar and the base. So we have polymers of DNA that exist as well. And here is an example of a polymer of DNA. And as you can tell, it goes from 5' to 3'. And the reason for that, it refers to the number of the carbon that we're referring to. So we always count this carbon as the number 1 carbon. This is like C1. So this is 1, 2, 3, 4. Here's the 5th carbon. This is why this is the 5' end. And the 3' end is, again, this is our C1 here. 1, 2, 3. This is our 3rd carbon, so this is a 3' end where the molecule kind of ends and stops there. So this is the 5' to 3'. And something really distinctive about DNA polymers is the existence of a phosphodiester bond. And I'm going to highlight that here for you so you can see it. But it's basically this phosphate group that connects the different nucleotides together. So this is that phosphate bond, or phosphodiester bond there. And it's actually considered a 3' to 5' phosphodiester bond. And the reason for this has to do, again, with the carbons that it attaches to. So here's our C1, then we have 2, 3, so here's our 3' carbon that's connected right there. Then we have 1, 2, 3, 4, oops, this is actually what I meant to draw, to this one. Here's our 5th carbon, so this is our 5. So this bond connects the 3' to 5' carbons here. And again, let me just draw it here. So here's our 3' carbon, here's our 5' carbon, and you see how it connects these two? This is our phosphodiester bond, and that's why it's a 3' to 5' phosphodiester bond. And then lastly, we're just going to go over the base pair DNA double helix. So some major characteristics of a double helix are that it's anti-parallel, meaning that the 3' and the 5' ends are corresponding and aligned with each other. So one goes 3' to 5', the other one goes 5' to 3'. The stability of a DNA molecule has to do with the presence of those hydrogen bonds between the bases, as well as something called pi stacking, which we talk about in the non-covalent forces video. So these two interactions is what really keeps DNA very, very stable and helps it pack also more closely together. And then we have the major and minor grooves. So here is a major groove, and then here is a minor groove, just something to do with the structure of the molecule. It's like smaller here between this one, which is bigger. Another really important thing to know is how many hydrogen bonds are formed between the bases. So adenine and thymine, which I'm sure bond together, they form 2 hydrogen bonds between the 2 bases, and guanine and cytosine form 3 hydrogen bonds between them. And the way I remember this is I just remember that G and C and 3 all rhyme together, and A and T don't really rhyme with 2, so I just tell myself GC3, and that's how I remember how many hydrogen bonds exist between them. And something I want to highlight also is something called Chargaff's rules, and this refers to the number of adenines and thymines and guanines and cytosines that you would expect in a DNA molecule. If you have, say, 30 adenines in your molecule of DNA, then you would expect that you would also have 30 thymines attached to it, because these 2 base pair together. And similarly, if you had 50 guanines in your DNA helix, then you would expect that there would be 50 cytosines, because these 2 base pair together. And since thymine and cytosine are let me draw this here. Thymine and cytosine are pyrimidines, and adenine and guanine are purines. You would expect that there's also a 1-to-1 ratio of purines to pyrimidines, because here we have 80 and 80. So this is just referring to the base pairings of the bases and how the numbers of them and the ratios of them that you would expect in a molecule based on the pairings between them. So that's an overview of DNA and the structure. So thank you for listening.